So hello everyone, welcome back to the session. So we are discussing about the CUET PG exam. So in that zoology particularly. So in that line we have discussed about the syllabus analysis and we have started the cell biology chapter and we have completed the cell theory and the prokaryotic and eukaryotic cell organization. So this is the third day of our class. In this class we are going to discuss completely about the plasma membrane with respect to the CUET exam, right? Okay, so fine. So first, what will be, how can we define this plasma membrane? Okay, so how can we define, this is the cell membrane. Okay, cell membrane is also called as the plasma membrane. So this is also the cell membrane is also called the cell surface membrane or the plasma membrane. Okay, cell membrane, cell surface membrane or the plasma membrane, everything denotes the same thing. So it is a thin, it is living and it is selectively permeable membrane. So this is very important statement. Okay, it is not completely permeable, it is selectively permeable. Okay, it is a selectively permeable membrane that encloses the cytoplasm. Okay, so this is the cell, this is the nucleus and these are all the cytoplasm, and the cytoplasm is covered by a membrane that is called as a plasma membrane. Okay, and the thickness of this plasma membrane is less than 10 nanometer. Okay, so what is the purpose of having the plasma membrane means? So it maintains the cell integrity, regulates the transport and enables the cell communication. One cell has to be communicated with the another cell. So to mediate the cell communication, there is a presence of this plasma membrane, and to maintain the cell integrity, to maintain a proper composition of a cell, we are having a plasma membrane. And it, as it is a selectively permeable, the contents which is present inside the cell that has been transport from one cell to another cell with the help of this plasma membrane. Okay, so these are all the importance of the plasma membrane, how this plasma membrane will look like. Okay, just to see this picture, these are all the model of the cell membrane. See, this is a, how can we, this is a head and this is the tail, this is water. This is what, one layer. Okay, so here another layer, so here there will be like this. Okay, so this is what, an lipid layer, and this is one lipid layer and another lipid layer. So we can say this plasma membrane is a lipid bilayer. Okay, so we are having a protein in the plasma membrane, we are having a glycoprotein, we are having a protein. And what is meant by this glycoprotein? This glyco means carbohydrate and this is a protein. Carbohydrate associated with the protein is also present in the plasma membrane, and there is a carbohydrate and there is a glycolipid. Glyco, it is a word, denotes the carbohydrate, and here a carbohydrate is associated with the lipid, so we can call them as a glycolipid. Okay, and there is a cholesterol. Okay, cholesterol is present in the layer. Okay, and see here, this is a lipid layer, this is one layer and this is another layer. Between these layers, the protein is integrated in the plasma membrane, so that we are calling them as an integral protein. Okay, now, and this is a lipid bilayer, and we are having a protein in the periphery region, so that we are calling this particular protein as a peripheral protein, and we are having a channel protein. We are having a channel protein so that all the contents has been easily transported with the help of this channel. Okay, channel, and this channel is nothing but a protein. It is made up of a protein, and also we are having an alpha helix protein. Okay, and this protein, we are having an alpha helix, beta pleated, these are all the structure of a protein, and we are also having the alpha helix protein in the plasma membrane. So finally, I just want to say that one important fact is the lipid is in higher concentration in a plasma membrane. Okay, and after that followed by a protein and followed by a carbohydrate. So this is an important proportion with respect to the plasma membrane. Okay, and this composition will be vary from one cell to the another cell. Say for an example, in case of a liver, there is a different percentage. Okay, and in case of RBC, that will be the different percentage of lipid, protein and carbohydrate. But majorly, lipid will always be in a higher proportion, and the protein will always be a higher, followed by a protein and followed by a carbohydrate. This is what a ratio. right? Now this is what a model of a plasma membrane. And what are all the chemical composition? Just now I have explained you. See, plasma membrane is a lipoproteinaceous structure. What is mean by lipoproteinaceous structure? This is made up of a lipid. See, plasma membrane is made up of a lipid and it is also made up of a protein. So lipid, lipoproteinaceous structure, and it is a right statement. And also see here, lipids, approximately 40 to 50% of lipids will be mainly phospholipids. Lipid will be present in the form of phospholipids. Okay. Now proteins, approximately 50 to 60% of proteins will be structural and functional protein. Okay. Now carbohydrates will be 50 to 10%, that will be present as a glycoprotein or it will be present as a glycolipids. Either the carbohydrate is associated with the protein or it will be associated with a lipid. Okay, mainly it is present in the outer surface. Okay, so what will be an exam focus? So when it comes to a CUET exam, how can we explain the composition of a plasma membrane? See, the composition varies among the cell types. Say for an example, in RBC cell membrane, there is 52% of protein and 40% of lipid. But what I have explained you before, lipid will always be higher proportion in the cell membrane, followed by a protein, followed by a carbohydrate. This is what I have explained you, but there is an exception, that is what an RBC membrane. Okay, so this may be vary with respect to a cell where it is presented and their function as well as their structure. Okay, so this is what a chemical composition of the plasma membrane. Now there is an important model which explains the structure of the plasma membrane, and that model is the fluid mosaic model. Okay, so this fluid mosaic model will be proposed by a scientist that is called as a Singer and Nicolson. Okay, Singer and Nicolson are the two major scientists. They proposed the fluid mosaic model in the year 1972. This is an important fact that you have to keep in mind. Okay, exam point of view, right? Next, this is a widely accepted model. So as we have as I have mentioned here as a widely accepted model, do they have any extra model? Of course, plasma membrane have many models proposed by different scientists. Okay, but the most accepted model will be the fluid mosaic model proposed by the Singer and Nicolson in 1972, right? So what this model explains, okay, this membrane consists of a phospholipid bilayer. As I have mentioned to you, this is a membrane, this is a lipid and this is a lipid, and there is a two layer of lipid. We can call them as a lipid bilayer, and the lipid will be in the form of phospholipid. So that is why we are saying a phospholipid bilayer. And also lipid with the proteins embedded in it. So what are all the proteins we have studied? We have studied peripheral proteins and we have studied integral proteins. So these are all the two important proteins. This is necessary. Now the lipids provide a fluidity and the proteins form a mosaic pattern. So that is why. So what is mean by this fluidity and what is this mosaic pattern? See, just listen to this picture. See this picture. So this is the lipid. Okay, so this is the top layer of the cell membrane and this is the bottom layer of the membrane. So like in the same way, there is a bottom layer also. So we are saying this lipid ensures a fluidity. Okay, and the proteins embedded in certain area. So that is why this is the protein resembles a mosaic pattern. So that is why the name fluid mosaic model. How can we, why it is fluid, why it is mosaic? The question can be, we can expect why with respect to the fluidity and the mosaic. Okay, see, why fluid? Why it has to be fluid? Lateral movement of the lipids and proteins within the bilayer. What is meant by this lateral movements? See, this is the lipid bilayer. So here we can see the lipid layer and here we can say the lipid. Okay, so here also we can say the lipid. So here if any particle that has to be moved laterally, Okay, so only if the plasma membrane is fluid, the movement, the lateral movement can be easily possible. So that is why there is a fluidity. Okay, why it has to be fluid? So if it is a fluid, the lateral movement of lipids and proteins will be possible. Okay, why it is mosaic? Irregular arrangement of proteins within the lipid matrix. Why we are saying this is a fluid mosaic model? There's a fluid ensures the movement, and the mosaic means the mosaic model enriches the arrangement of a protein. So this is why we are calling them as a fluid mosaic model proposed by a Singer and Nicolson in 1872. All these are an important statement we can expect in an examination. Okay. Now, this is how a plasma membrane will look. Okay, see, this is what a phospholipid layer. Okay, so again it is a phospholipid layer, so that we can say the phospholipid bilayer, and we are having a peripheral proteins here, peripheral proteins, and this is a sugar, which we can say this as a glycoprotein. Okay, and here also we can say the layer, and at certain area we can see there is a cholesterol which is embedded in it. Okay, so all these are important thing. Okay, so first thing, phospholipid bilayer organization. So as I have mentioned you before, there is a phospholipid bilayer. This is a head region and this is a tail region. Okay, see, each phospholipid, so this is one phospholipid, they are having a head region and they are having a tail region. Okay, head region and the tail region. Okay, so this head region is called as a polar. Polar means that attracts water, so that it is water loving, so that we are calling this particular head as a hydrophilic head. Okay, so this is a non-polar. The tail region is called as a non-polar region, non-polar tail region. We are calling this particular tail as a hydrophobic, as it is water hating. Okay, it repels water, it attracts water. So the important fact that you have to keep in mind is the head region. The head region is hydrophilic, polar, attracts water, and the tail region is it repels water, it is non-polar, it is hydrophobic. These are all the important factual information you have to keep in mind. Okay, see, the polar head faces the aqueous environment and both the sides. See, this is the head region. This is the head region on both the sides. Again, here also we can see the head region. Both the polar head faces the aqueous environment on both the sides. We have studied the lipid bilayer. On the upper side also we can see the head region, and the lower side also we can see the head region, and both the heads faces the water environment, that is aqueous environment on both the sides. And this hydrophobic tail faces inward. So this is very, very, very important. The question may be asked whether the hydrophobic tail face inwards or outwards. You should know that hydrophobic tails face inwards, hydrophilic head faces outwards. Okay, faces the aqueous environment on both the sides. You have to keep in mind. Okay. An important point will be this lipid bilayer ensures a stability of the membrane in the aqueous environment. So this is an important factual information. Why it has to be lipid bilayer. Okay. Now, so another important thing will be the membrane protein. So we have studied about the phospholipids. Okay. Now the membrane protein. So membrane proteins, it is based on the ease of extraction and the position. Based on the extraction and where it is located, we have categorized the proteins into two. Okay, I repeat, based on the extraction process, based on how easily these proteins can be extracted and the position where it is located on the plasma membrane, we can categorize the membrane proteins into two. And they were the integral proteins and peripheral proteins. See, the integral proteins, we can call this particular integral proteins as intrinsic protein and this peripheral proteins as extrinsic protein. See, this is a lipid bilayer. As this is a lipid bilayer, these proteins are integrated into the lipid bilayer. As it is integrated in the lipid layer, we can say this particular thing as a integral protein. See, this is a lipid bilayer. As this is a lipid bilayer, the protein which is integrated completely in the membrane, we are calling them as a integral proteins. Now, what is with respect to the peripheral protein? The peripheral proteins will be attached like this. Okay, so when you look at these two types of proteins, which proteins can be easily extracted from this plasma membrane? See, you just by changing the pH, we can easily remove, we can easily extract the peripheral protein, but we have to use certain detergents. Okay, only then we can remove the integral protein. So you should know peripheral proteins are very easy to extract from the plasma membrane when compared to the integral proteins. So these are all very important points. See, the integral proteins embedded partially or completely in the lipid bilayer. Okay, as I have mentioned you here. Mostly project beyond the bilayer. This integral proteins are the proteins. This is the one act as a channels, act as a carriers, act as an enzymes, act as a receptors, act as an antigen. So these are all the integral proteins. Now what is meant by this peripheral proteins or extrinsic proteins? These are loosely attached on the membrane surface so that we can easily extract the peripheral proteins from the plasma membrane. Now, what is the function of these peripheral proteins? They provide the structural support and they are involving in the cell signaling process. Okay, so this is very important. Now we have studied about the lipid and we have studied about the protein. Now comes to the carbohydrates. Okay, membrane carbohydrates. Now glycocalyx. Okay, see, carbohydrates are short chain polysaccharide, short chain polysaccharide. They present only on the outer surface of the membrane, not in the inner surface of the membrane. This carbohydrates will not associated in the membrane, but instead it will be associated with the glycoproteins and glycolipids. Okay, so together they can form the glycocalyx. Now what is the function of this carbohydrates in the plasma membrane? They are important in the cell recognition and immunity. So this is very important. Now membrane dynamics. Now what are all the process which is happening? What are all the mechanisms happening in the membrane? See, they are having a lateral diffusion, flip-flop movement, phospholipids, lateral diffusion and the flip-flop movement. See, as I have mentioned it before, there is a difference between this lateral diffusion and flip-flop movement. See here, this is a lipid bilayer. Okay, lateral diffusion means some substances has been diffused laterally like this sidewards manner. Okay, now which means the lipid bilayer. Okay, so now this is a flip-flop movement. See, this is a flip-flop movement. This is a lipid bilayer. The entire layer will be like this. Okay, the entire layer will be like this. Now this region will be flip into downwards. So this is sidewards lateral movement, and here this the flip-flop movement means it will be flip here. Okay, the vertical movement. So it will be flip like this. So this is what a flip-flop movement. Lateral diffusion means side to side movement of lipids. This is a fast movement. And the flip-flop movement, this is a vertical movement, which means suddenly it will be flip into downwards. This is what a vertical movement of a lipids, and this is very slow. For this flip-flop movement, definitely it means energy. Okay. Now, see here, the phospholipids can flip-flop due to the smaller polar heads. The proteins cannot flip-flop due to large polar regions. Okay, see here, the phospholipids can flip-flop. Why? Because they are having a small polar heads in it. And the proteins cannot flip-flop. Why? Because due to larger polar region. So this is what a main reason why the lipids are involving in the flip-flop movement, why the proteins cannot. Okay. Now, what will be the role of cholesterol in the plasma membrane? Say they present mainly in the animal cell membrane. Okay, the cholesterol plays a crucial role in the cell membrane. Mainly it is present in the animal cell. This cholesterol regulates the membrane fluidity. The fluidity is very important, and that has been ensured and regulated by the cholesterol. Okay, they are increasing the rigidity at a higher temperature. Okay, at the high temperature they increases the rigidity. At low temperature they prevents the solidification. This is very important. Okay, see, at the high temperature, okay, at the high temperature the fluidity will be high. Just to note this point, if temperature is high, the fluidity will also be high. Okay, if temperature is low, the fluidity will be less, and the membrane will be solidified. Okay, which means they will be solidified. It reduces the fluidity, it increases the rigidity, so that no movement can be that much efficient. Okay, so at higher temperature, they increases the rigidity. At that time they are reducing the fluidity. Okay, at low temperature they are increasing the fluidity, they are reducing the rigidity, they are reducing the solidification. So this is very, very, very important. Just kindly make a note of that. Okay, now, so now come to the another important thing. As a plasma membrane, it will be selectively permeable. Okay, the selective permeability, in the sense, this plasma membrane allows a selective movement of the substances. The plasma membrane will not allow all the substances, but instead it will allow some important, what are all the substance which is necessary for the cell. Okay, that is why we are saying selective permeability. Now, say for an example, those substances, those molecules which are very small and non-polar molecules, they can pass easily. That is oxygen and carbon dioxide, they can pass easily. Okay, the large or charged molecules, they require the transport proteins. They require the transport proteins only with the help of this transport proteins like channel or carrier, only then this large or the charged molecules can pass through. Okay, but this oxygen and the carbon dioxide, that is the small non-polar molecules, can diffuse easily. Okay, now that depends on the lipid solubility, size of the molecule, the presence of the transport proteins, which means the selective permeability which depends on the lipid solubility, size of the molecule, and the presence of the transport protein. So this is very important. Now, how the transport occurs in the plasma membrane. Okay, we have studied about the transport proteins. So what are all the transport proteins? So we can classify them into two. Okay, passive transport and active transport. The basic difference between the passive transport and active transport will be with the use of energy, without the use of energy. Passive transport, in the sense, they don't need energy. Okay, active transport means they need energy in the form of ATP, so that it can transport the substances. Just come to the point. See, passive transport, they don't need ATP. It occurs along the concentration gradient. Okay, say for an example, what are all the types of the passive transport? Simple diffusion, facilitated diffusion, osmosis. These are all the three important types coming under the passive transport. All these transport doesn't need energy. The first thing will be the simple diffusion. It simply crosses the membrane, as because it is very smaller in size, like gases. Okay, so gases, lipid soluble molecules. See, very important here. If that molecule is made up of a protein, it cannot easily pass through the membrane. If the molecules is made up of the lipid, it can be easily cross the membrane. Why? Because the plasma membrane is made up of lipid bilayer. As it is made up of a lipid bilayer, the lipid soluble molecules can be easily cross the lipid bilayer. If the molecules is made up of a protein, if it is a proteinaceous in nature, this membrane cannot be this molecules cannot be crossed the plasma membrane. So this is the very important thing. And the second thing will be the facilitated diffusion. See, facilitated diffusion, this also doesn't need energy, but instead they are carrying the molecule with the help of the carrier or the channel proteins. Someone is carrying the molecule and it is easily crossing the membrane. What are all the carrier or channel protein? That may be specific or that may be saturable. So this is also a facilitated diffusion. Now the third important transport will be the osmosis. Osmosis, in the sense, through the semi-permeable membrane, there will be the diffusion. The diffusion occurs through the semi-permeable membrane. See, another important pinpoint will be this. The facilitated diffusion is specific, but it is energy independent. Of course, it will be specific, but it doesn't need energy. But instead, it will be carried by someone. It will be facilitated by someone, and it will be easily carrying the molecule. This is what the passive transport. Now comes to the active transport. Active transport, in the sense, they definitely need energy. with the help of in the form of ATP. That's why we are saying this is an ATP dependent transport. Okay. It occurs against the concentration gradient. See, passive transport occurs along the concentration gradient. Say for an example, so this is a water molecule and this is the boat. Okay, this is the boat. The waves occur, the wind flows from this side to this side. Okay, so if the ship is go from this side to this side, they didn't want to give in extra effort. Okay, just with the flow of wind the ship can easily move. But remember, if the wind is flowing like this and the ship has to go from this side to this side, this is against the concentration gradient, so definitely we have to put some extra effort. So this is what an active transport and passive transport. Okay, now, it occurs against the concentration gradient and it uses the carrier protein. Say for an example, sodium potassium pump. Okay, this maintains an ionic balance and membrane potential. Okay, so this is what an active transport. Now comes to the bulk or vesicular transport. Okay, that is endocytosis, exocytosis. Endocytosis means some materials that has to be entered into the cell. What are all the types? Okay, so what are all the types of endocytosis? Endocyte means the cells, they are engulfing the material bulkily in the form of vesicles. Okay, so they are pinocytosis and phagocytosis. If it is a liquids, then pinocytosis, the process name will be the pinocytosis. If it is a solid, that process will be phagocytosis. It is simply the cell is engulfing the substances. Okay, this is absent in the plant cell due to rigid cell wall. So this is very important. Another important common name for the pinocytosis will be cell drinking. Okay, now exocytosis, removal of or secretion of materials. This is important in the hormone secretion and neurotransmitter release. Okay, see, some of the bulk material that has to be sent out of the cell, so that process is exocytosis, and this is important in case of hormone secretion and neurotransmitter release. This is what a bulk vesicular transport. Now what are all the functions of this plasma membrane? Why the plasma membrane has to be present in the animal cell? Okay, see, it is the one maintaining a cell shape. This is the one regulating the transport. This is the one enables the cell signaling and provides anchorage for cytoskeleton. Anchorage means support. Cytoskeleton, they are nothing but microtubules, microfilaments, intermediate filaments. All these things will be discussed detailing in the cell division or cell cycle chapter. Okay, so this is involving in cell adhesion, cell attachment. Okay, so these are all the functions of the plasma membrane, and these are all the important factual one-liner. Okay, so fluid mosaic model was proposed by Singer and Nicolson, and the plasma membrane is asymmetrical. Why it is asymmetrical? We have studied. There is a higher concentration of lipid and followed by protein, followed by carbohydrate. It is not in an equal proportion, so that is why it is asymmetrical. And here, facilitated diffusion not equal to active transport. What do we have discussed? Passive transport means we need no energy. Active transport means definitely we need energy. Okay, we have different types of passive transport, that is simple diffusion, facilitated diffusion, osmosis. Okay, so facilitated diffusion is specific but it is energy independent. So that facilitated diffusion, it is not equal to active transport. So kindly keep in mind. Okay, endocytosis occurs only in animal cells. So this is an important thing. Okay, so we have discussed this before. And the cholesterol, what is the point of having the cholesterol in the plasma membrane? So this is the one stabilizing. In case of higher temperature, they are reducing the fluidity. In case of lower temperature, they are increasing the fluidity. So this is what maintained, stabilized by the cholesterol. Okay, so now another important traps that the exam can trap you. Okay, so the plasma membrane is rigid, so this is very false. Okay, just now we have discussed it should not be too rigid, it's not too much fluid. Okay, it has to maintain in balance. All transport requires ATP. So just now we have studied some may not need energy. So this statement is also false. Carbohydrates present on both sides. So we have studied carbohydrates only present on the outer side. So this statement is also a false statement. Okay. So now so we have completed the most important things with respect to the plasma membrane. Now comes to the MCQs. Okay. The plasma membrane is best described as, take 1 minute of time or just pause the video and just answer the question. Okay. The plasma membrane is best described as a rigid, a non-living covering, selectively permeable living membrane, a porous carbohydrate layer, a double protein layer. So first thing, it is not rigid. Okay, it is not non-living, it is in living. Okay, it is not porous carbohydrate layer, it is not a double protein layer, but instead it will be selectively permeable, it is living, it is made up of lipid bilayer. So the correct answer will be option B. Okay, the second question will be, the fluid mosaic model of the plasma membrane was proposed in 1972 by Singer and Nicolson. So this is the right answer. Okay, and the third question will be, which component of the plasma membrane is mainly responsible for selectively permeable? Okay, selective permeability. Who is responsible for this? So which component? Carbohydrates, proteins, cholesterol and phospholipid bilayer. So there might be a confusion, but the answer will be, there is the hydrophobic core of the phospholipid bilayer restricts the polar nucleus. So the correct answer will be option D, that is the phospholipid bilayer is the one responsible for the selective permeability. Okay. Now, the fourth question will be, the facilitated diffusion differs from the simple diffusion. So, before that you should know, both facilitated and the simple diffusion are the types of the passive transport, but you should know what will be the difference. Okay, simple diffusion doesn't need energy. Of course, facilitated diffusion also doesn't need energy. So it requires ATP. Of course, it is not. Occurs against the concentration gradient. No, again it is not. Uses specific carrier proteins. This is the right statement. It's restricted to ions only. No, it is not a right statement. Okay. So the correct answer will be option C. Now, the endocytosis is absent in plant cells mainly due to the presence of. Why the endocytosis is absent in plant cells, but only the endocytosis will be present only in the, only in the animal cells. Why is that so? Why? Because there is a rigid cell wall. So this is the one preventing the membrane invagination. Now, carbohydrates of plasma membrane are located mainly on the, where exactly the carbohydrates of the plasma membrane is located, whether it will be in the inner surface or both surface or outer surface or the lipid core. So the answer will be outer surface, as I have mentioned you before. Now, the flip-flop movement across the plasma membrane is shown mainly by, so in the plasma membrane we are having a lipid, we are having a protein, we are having a carbohydrate. When compared to the lipid, protein and the carbohydrate, who perform this flip-flop movement? Who is undergo this movement? Integral proteins, peripheral proteins, phospholipids and glycoproteins. All these were the proteins. All these were the proteins. The proteins cannot show this flip-flop movement. Why? Because larger polar heads. Okay. But this phospholipids is the one can undergo this flip-flop movement. Why? Because smaller polar heads in it. Okay. Now, the eighth question will be, which transport process moves the substance against the concentration gradient? Against the concentration gradient in the sense, simple diffusion, facilitated diffusion, osmosis. All these were the types of the passive transport. It doesn't need any energy. Why it doesn't need an energy? Because they transport, they move the substance along the concentration gradient. So the only thing is, the active transport is the one process, the movement process moves the transport across the concentration gradient. So the answer will be option D. Now, the ninth question will be, the cholesterol is an important component of an animal cell membranes because it, okay. So why it is important as a component of the animal membrane? Animal cell membranes. Why? Because it is the one regulating the membrane fluidity, acting as a receptor, ion channel, provide energy. This is irrelevant to the question. So the answer will be regulating the membrane fluidity. Now comes the 10th question. Which statement about the plasma membrane is incorrect? It is asymmetrical. Again, it right. We have studied this before. Proteins can move laterally and it is selectively permeable. Of course, it is right. It is rigid and static structure. So it is rigid as well as it is dynamic structure. Of course, it is right. So the answer will be. I'm sorry. Proteins can move laterally, selectively permeable. It is not rigid. It is fluid. Okay. So it is a fluid in nature. So that's why. So the word you should not confuse with by seeing rigid as well as static. Okay. It is fluid in nature. So that the answer will be option D. Okay. So in today's class we have completed about the plasma membrane. We'll meet you in the next class. Okay. Thank you so much.